Hello everyone, very good evening to all. This is Dr. Abirami, your physiology faculty. So we'll just wait for 2 more minutes. Students are just joining. Just 2 more minutes we'll wait and we'll start the session. Okay, students are still joining. We're about to start the session. Okay, so I think we can start our session. So I welcome you all to DBMCI MDS live regular batch physiology sessions. So we are starting with physiology sessions, and just for a good start of the class, usually I start my classes with this small story or something which happened really. Have you seen this program called The Millionaire? Yes, it's an English program where the person who's conducting the program will ask you 20 questions and you answer all the questions. You will have a chance to win $1 million. That is about the program. And this man, John Carpenter, he was the first person to win $1 million in this program. And how he won this was really interesting. There are a few lifelines in the program, like you can call somebody and you can ask the answer. It's a live program telecasted all over the world. Everybody is watching, but on live you can call somebody and you can ask the answer if you don't know the answer. And there are options like 50-50. Out of the 4 options, 2 options will be removed. So like this, there are few lifelines, and this man, he did not use any of his lifelines till his last question. And on the last question he said, I want to call somebody. So he said, I want to call my father. It's a live program. Everybody is watching. He's calling his father and he's saying, Dad, I did not call you to ask the answer for any question, but I just wanted to tell you that I'm going to win $1 million in the next question. He kept the phone, he gave the right answer, and he won $1 million. This much of self-confidence. How will you get this much of self-confidence? Even without knowing if it is the right answer or not, he's just telling to his father that I'm going to win $1 million in the next question. This much of self-confidence. Only with clarity in the subject you will gain this much of self-confidence. When you go for the exams, students keep searching for self-confidence everywhere, in a WhatsApp message or in a Facebook video or in an Instagram reel where somebody is climbing on a mountain. There's a very motivating BGM behind. You will not get self-confidence from any of these sources. Instead, you sit and learn your subjects. If you have clarity in your subjects, that will give you enough self-confidence. You learn physiology. You go to the exam, you see the first question on physiology. If you know the answer for the first question, this will give you the confidence for the second question. If the first 10 questions are right, that will give you the confidence for the entire paper. If the first question or the first 10 questions are wrong, you don't know, you will lose your confidence in the exam. So what is exactly giving you the confidence? Clarity in the subject. So if you want to gain enough self-confidence before going to the exam, have enough preparation, learn all the subjects thoroughly, be clear in all your concepts, just go to the exam, you will know the answers for all the questions. So that was about John Carpenter. The importance of basic subjects, especially physiology. Physiology forms a strong basis for three other clinical subjects: medicine, pathology, and pharmacology. As such, in physiology, what is the weightage of questions that are asked? Approximately around 10 to 13 questions you can expect with physiological basis. Only if you know physiology, you will be able to get through these 13 questions that are asked in from underlying basics of physiology. So that is the power of learning basic subjects. And moreover, once you learn physiology, then learning clinical subjects like medicine, pathology, and pharmacology becomes very, very simple for you. For example, there is a question on ECG in medicine, a clinical question. How will you answer this question without knowing the basics of ECG in physiology? Yes. There is a question on insulin in pharmacology. Without knowing what is insulin, what are the functions of insulin, how will you be able to answer this question? So that is why learning physiology is really very, very important. So this is about learning the importance of basic subjects in MDS exams. Over to books that you read. Usually students keep asking the book of choice for physiology entrance preparation. As such, there is no one book of choice. There are many books from which questions can be asked, but if you ask me the Bible to physiology, definitely it is Genome. Most of the questions or all of the questions in physiology are asked from this book, Genome's Review of Medical Physiology. But like this, you have 20 other textbooks, standard textbooks for 20 other subjects. It is impossible for you to sit and read all these books. You can't sit and read Genome during your 6 or 7 months of preparation. You need at least 1 month to read Genome itself. So that is the essence of attending our classes. You attend my physiology class, I am going to give you a 25 to 30 hours of extract from Genome. So whatever is there in Genome, so I have concised it, made it into the class, and that is what I am going to deliver you. So that is the power of learning from the expert faculty, where we are going to give you the extract of all standard subjects. Likewise for every other subject also. So that was about a small introduction to our class. Now let's start off with the first chapter, General Physiology. General Physiology, very basic chapter. We have many basic clarifications in general physiology which will help you to understand systemic physiology better. So that is why we start with general physiology now. Okay. Who is this person in general physiology? There are two people here. This man. Look at this man. Can you answer me this question? Yes, very good evening, Dr. Aman Chaudhary, Dr. Parvati, Harshal, Dr. Libra. Very good evening to all. Can you tell me whose father is this? Yes, Dr. Parvati is there. Libra. Can you tell me whose father is this? Yes, whose father is this? Can anybody tell me whose father is this? If I am asking you the question, not my father, whose father is this? Yes, father of physiology. Who is called father of physiology? Claude Bernard is called the father of physiology. Because of his important contribution, this term called milieu interior. What is milieu interior? The internal environment of the body is called milieu interior. What is the internal environment of the body? ECF, extracellular fluid. Claude Bernard gave this term milieu interior to the extracellular fluid, which means ECF is the internal environment of the body on which all cells are embedded. So that was about Claude Bernard. Yes, exactly, Dr. Yatra and Monica, father of physiology. Let me introduce you to another person, an important scientist related to general physiology. Yes, he is Walter Bradford Kennan. W.B. Kennan. What are the contributions of Kennan? Kennan coined this term, homeostasis. What is homeostasis? Maintaining the constancy. To maintain the constancy of the internal environment of the body. That is called homeostasis. What are the examples for homeostasis? How you maintain your body temperature, your blood pressure, blood pH, blood gases, acids, bases, electrolytes. All this is homeostasis. All these are examples for homeostasis. And the other thing I wanted to tell you, if you are not able to take notes of these classes, so we will be sharing the PDF of these notes. Don't worry about that. So after the end of all the 25 hours of classes, because we won't have time to, you won't have time to write down all these. So I'll share the PDF of all these notes. Don't worry. Let's move on to components of homeostasis. What are the important components of homeostasis? Whenever there is any imbalance in homeostasis, that is the stimulus which is detected. Who will detect this? The receptors. The change is detected by the receptors and they will send afferent pathways into the control centers. From the control centers, you have efferent pathways arising to reach the effector organ, and finally, as a response to this change, the imbalance is corrected. For example, there is any imbalance in blood pressure. If there is any rise in BP detected by your baroreceptors, they reach the center, nucleus tractus solitarius of your brain, efferents come back to the BP centers, and that is how there is a fall in BP. So this is called baroreceptor reflex, which is an example for maintaining homeostasis. Over to control systems. What are the control systems which will maintain the homeostasis? Who is going to maintain all this homeostasis? Control system. What are the control systems? We have two types of control systems: feedback control system and feed forward control system. What is feedback control system? Let's do that first. There are two types of feedback control system: positive feedback and negative feedback. What is a positive feedback? When there is a stimulus and a response, stimulus increases the response and response increases the stimulus. One is enhancing the effect of the other. This is a vicious cycle. This is an example for positive feedback. What about negative feedback? In negative feedback, there is a stimulus and a response. Some disturbance in background sound. Okay, somebody saying there's disturbance in background sound. Let me check that. Yes, now is it fine? Okay. If the stimulus and the response are acting in opposite directions, this is example for negative feedback. For example, if there is an increase in T3 and T4 levels, what will happen to TSH? TSH decreases. Or vice versa also. If there is an increase in T3, T4, there is a decrease in TSH. If there is a decrease in T3, T4, there will be an increase in TSH. So this is an example for negative feedback. What are the other examples for negative feedback? Basically, all hormone mechanisms, they follow negative feedback except two. Can you say what are those two? Except two. What are those two exceptions? Exactly, Dr. Rahul Chandrasekhar Bilur. Very good. Hyperthyroidism and hypothyroidism. Exactly. What are those two exceptions which do not follow negative feedback? Anybody? Yes, it is LH and oxytocin, luteinizing hormone and oxytocin. All endocrine hormones follow negative feedback except luteinizing hormone and oxytocin, which follows positive feedback. Another example for negative feedback: blood pressure regulation by baroreceptor mechanism. Whenever there is a rise in BP, baroreceptors would decrease the BP. Yes, exactly, Dr. Rahul and Yatra. Very good. So all these are examples for negative feedback. What about positive feedback? In positive feedback, the mnemonic is CLAPS. C L A P S. C for clotting mechanism. What happens in clotting mechanism? If this is a blood vessel which has been injured and this attracts platelets, platelets release chemical mediators, they in turn attract platelets and again they release chemical mediators. This is a vicious cycle. One is enhancing the effect of the other. That is how clotting is an example for positive feedback. Another C for calcium induced calcium release. This is in the cardiac fibers in your heart muscles. There is calcium induced calcium release. Entry of calcium releases calcium. In CVS we will learn this in detail. L for LH surge. This happens exactly before ovulation. There is an LH surge. What is LH surge? On the 14th day there is ovulation and exactly just before ovulation there is LH surge. Increase in LH. This increases estrogen and estrogen further increases LH. LH again increases estrogen. This is also a vicious cycle. One is enhancing the effect of the other. And another L for lactation reflex. What is the other name for lactation reflex? Milk ejection reflex. Lactation reflex or milk ejection reflex. What is lactation reflex or milk ejection reflex? If this is the mother and this is the baby, the more the baby sucks milk, more the love of the mother increases and the mother starts to produce love hormone. Which is the love hormone? Oxytocin. What is the action of oxytocin on breast cells? Oxytocin further increases contraction of the myoepithelial cells of your breast and therefore more milk is ejected. More the milk ejected, more the baby sucks. More the baby sucks, love of the mother increases, more the love hormone, again it causes contraction of the breast and hence more milk is ejected. So this is a vicious cycle. One is enhancing the effect of the other. And that is why oxytocin we call it as a love hormone. Only if the love of the mother for the baby increases, breastfeeding, normal breastfeeding or lactation is possible. If the mother is not willing for breastfeeding, you can't feed the baby. A for action potential. This we will discuss in detail in nerve physiology about action potential. P for parturition reflex, which is otherwise called Ferguson's reflex. Parturition reflex or Ferguson reflex. What is parturition? At the time of delivery of the baby. So something similar to lactation reflex. I'll tell you what is parturition reflex. This is the mother and uterus of the mother. More the uterine contraction, more the baby descends down. At the time of delivery of the baby, more the baby descends down, love of the mother increases and mother produces love hormone oxytocin. Oxytocin causes increased contraction of the breast. Again the baby descends down. Here it is uterus, increased contraction of the uterus. Again the baby descends down. More the baby descends down, the love of the mother increases. Mother produces oxytocin. Again oxytocin causes contraction of the uterus. And this is a vicious cycle. It keeps on happening until the baby is delivered. So this is again an example for positive feedback mechanism. S for shock. What is shock? This is not the electric shock. When body is in shock. What happens when body is in shock? Heart is not pumping. When heart is not pumping, cardiac output decreases. When cardiac output decreases, BP decreases. When BP decreases, venous return decreases. When venous return decreases, again cardiac output decreases, BP decreases, venous return decreases. This is again a vicious cycle. It will keep on happening until you intervene and treat the patient. If you fail to intervene and treat the patient will die from shock. So CLAPS, CLAPS. This is a mnemonic for examples for positive feedback mechanism. So we have learnt about feedback systems. Now let's learn about feed forward systems. And oxytocin is called love hormone. Do you know that? Why oxytocin is called love hormone? Only if the love of the mother for the baby increases, breastfeeding is possible, normal delivery is possible. Not only the love of the mother for the baby. Wherever there is love, there is oxytocin. There is a Russian study which did a research, and then only they proved that oxytocin is the love hormone. What was that study? They measured the levels of oxytocin in 100 couples in different parts of life. For example, they measured the oxytocin levels just before marriage. People are actively in love, so oxytocin levels are very high. And just after marriage, immediately after marriage, now also the levels were high. Now only they've got married, right? So still people are in love. Measure the levels of oxytocin after 1 year, slowly decreasing, 2 years, 3 years, and finally oxytocin vanishes. So researchers found out that when really people are actively in love, oxytocin levels are high. So if you want to check your love, if you are in love, please go to the lab, check your oxytocin levels, it should be high. Or if you want to check your partner's love also, take them to the lab, check their oxytocin levels, it should be high. And hence we named it as love hormone. So that was about feedback control system. Over to feed forward control system. What is feed forward control system? In feedback, we had a stimulus and we had a response. But in feed forward, there is no actual stimulus, but still there is a response. Without actual stimulus, how will you get a response? Yes, sometimes we do get a response, and these are examples for feed forward system. First example, anticipatory tachycardia. Anticipatory means you predict beforehand itself. That is anticipatory. What is anticipatory tachycardia? When you run on a treadmill, your heart rate increases. That is tachycardia. But sometimes, even before getting onto a treadmill, your heart rate can increase. How will this happen? For example, you take your grandparents near the treadmill and you just tell them you have to run for the next 1 hour on full speed on the treadmill. Even before getting onto the treadmill, their heart rate will increase. So just thinking about that event itself, there is a response. So it is an anticipatory tachycardia. And number 2, cephalic phase of gastric acid secretion. What is the stimulus for gastric acid secretion? Food only. Food is the stimulus. Sometimes even without food, thinking about the food, or if I bring a nice plate of biryani near your nose and you smell that food, just by thinking or smell of the food itself can increase acid production. This is called cephalic phase of gastric acid secretion. And number 3, posterior hypothalamus produces shivering. If you enter into a cold room, your posterior hypothalamus will detect that you have entered into a cold room. It is very cold there. So before your body temperature could fall, it will make you to shiver. Why do you shiver? When you shiver, your muscles are contracting. When your muscles are actively contracting, what will happen if muscles are contracting? More heat is produced. So that is how when you enter into a cold environment itself, your body is making you to shiver. And number 4, motor correction by cerebellum. This is the cerebellum. And what does the cerebellum do? If suppose you miss your steps, you don't actually fall, you just skip your steps, you miss your steps, your cerebellum will detect that you're going to fall, and even before you could fall, it will give you a correction. And that is how every time we miss the steps, we don't fall. Somehow we balance and we come into normal position. And number 5, insulin secretion by pancreas. Pancreas produces insulin in response to blood glucose. Whenever your blood glucose level increases, pancreas can produce insulin. But sometimes, even before your blood glucose could rise, you have just eaten a full plate of biryani, it is full of carbohydrate. When you eat carbohydrate itself, your pancreas will detect that he's eaten nice carbohydrates. It is anyway going to be converted into glucose. So let me start producing insulin now itself. So even before your blood glucose could rise, just after you eat a carbohydrate rich food itself, insulin secretion can increase. So that is an example for feed forward control system. So all these are examples for feed forward control systems. Over to gain of a system. We had learned about few of the control systems. How will you assess that any control system is working well or not? You calculate gain of the system. If the system has a good gain, it means it is effective. If the gain is not good, it is a very bad system. What is the formula to calculate gain? Gain is equal to correction divided by error. How will you remember this formula? You would have done lot of errors in your life. Of all the errors, how much ever you have corrected is a gain for you. Put all your mistakes down. Of all your mistakes, of all your errors, how much ever correction you have done is a gain for you. That is how gain is equal to correction divided by error. There is one system which has an error of 0. It means the system has no error at all. If any system has no error at all, how much will be the gain? Gain is equal to infinity. Yes, if there is no error, you will have infinite gain. Which is that only system which has error of 0 and therefore gain of infinity? Renal regulation of blood pressure. When your kidneys are regulating blood pressure, gain is infinity because error left by the system is 0. This is how we calculate gain of a system. Let me show you a question. Just see if you are able to calculate the gain of the system using the formula. Gain is equal to correction divided by error. How much is the correction of the system? On changing position from lying down to standing, there is a drop in 10 mm of mercury pressure. Immediately he recovers by 8. There was a 10 mm of mercury blood pressure drop, but he has recovered by 8, leaving behind 2. 8 has been corrected. 2 is the error left by the system. Of the 10 mm of mercury fall in BP, 8 has been corrected. So correction is 8. Error is 2. Now what is the answer? 4. So the answer is 4 to this question. Over to body fluid compartments. Body is made up of solids and fluids. How is the body fluid compartment again subdivided as? First let's see the solid compartment. So the top compartment, I'll shade that in blue color. Yes, this is the solid compartment. And body is made up of water. We call it as the total body water compartment. So in green, I'll shade that in green color. So whatever I'm shading in green, this is the total body water compartment. How is this total body water distributed in any person? It depends on males, females and infants. In any female, there is 55% of total body water. In any male, 60% of total body water. In infants, 75% of total body water. This is how total body water is divided in any person. Now again, these compartments can be subdivided. For any example, we usually take an adult male of 70 kg. So let me take an adult male of 70 kg. If this is any adult male of 70 kg who has 60% of total body water. How is this? 60% of total body water distributed in any adult male. See, this 60% of 70 kg is fixed. If it is an 80 kg male, it would be 60% of 80 kg. If it is a 90 kg male, it would be 60% of 90 kg. Now let's see how is this total body water distributed. Where will water go inside the body? Water goes into the cells inside the body. So this we call it as the intracellular compartment. And the remaining water is distributed in the extracellular compartment. This we call it as the ECF, ICF and ECF. Which you think will be more, inside the cell or outside the cell? We generally think that outside the cell the space only will be more, but actually body is made up of millions and millions of cells very closely packed like this. So overall, if you calculate, the outside space only will be more. So when you compare intracellular fluid and extracellular fluid, when you compare intracellular fluid and extracellular fluid, inside space only will be more, not the extracellular. I said ECF, no, it's not ECF, it is ICF. Overall, if you compare, since body is made up of millions and millions of cells very closely packed like this, the intracellular compartment only will be more. So we'll do that. The total distribution of total body water is like intracellular compartment. This occupies 2/3 of the fluid. So 2/3 goes to ICF. 2/3 means 40% goes to ICF and only 1/3 goes to ECF. 1/3 ECF. 1/3 is 20%. Of 60%, 2/3 is 40% and 1/3 is 20%. Again, ECF has two other compartments. What do you have outside the cells in the ECF? We have the space. The space is called interstitium, and we have blood vessels. Blood vessels contain plasma in them. So outside the cells in the ECF compartment, again we have two divisions, interstitium and plasma. How is this distributed? 20% is distributed as 15% interstitium and 5% plasma. In terms of numerical values, let's do a calculation. How much do you think is 60% of 70 kg? Yes, 42 liter. 60% of 70 kg. Excellent, Dr. Agarwal, Muskan Agarwal and Rahul Chandrasekhar. Very good for the answers. How much is 60% of 70 kg? 60% of 70 kg will be 42 liters. So it is 42 liter. Again, this 42 liter is divided as 40% and 20%. So this would come up to 28 liter ECF and 14 liter 28 liter ICF and 14 liter ECF. Again, this 14 liter is distributed as 10 liter interstitium and 4 liter of plasma. This is how total body water is distributed in any adult male of 70 kg. This is only for 70 kg. According to the weight, all this will change. How do you measure all these compartments? What is that technique used to measure body fluid compartments? It is called indicator dye dilution technique. Using this technique, we inject specific dyes into the patient. It depends on the property of the dye. If that dye has the property of occupying the total body water compartment, you inject that dye and you measure the volume of distribution of that dye. That will tell you how much is the total body water. Likewise, when you want to measure ECF, you inject a particular dye. If that dye has the property of occupying ECF compartment, that will tell you how much is the volume of distribution of ECF. So likewise, we have different dyes for different body fluid compartments. For measuring total body water, I can use something which is like water itself. Heavy water? Yes. For measuring total body water, I can use heavy water, which is called deuterium, tritium, antipyrine, aminopyrine. So all this occupies total body water compartment. For measuring ECF, extracellular fluid, I can use something which is more concentrated in the ECF, which is that ion which is more concentrated in the ECF, sodium. So I can use something which is like sodium. I can use sodium thiocyanate, sodium thiosulfate, radioactive sodium, mannitol, sucrose, inulin. That's why ECF is otherwise called as sucrose space or inulin space. All these substances can be used to measure the ECF compartment. For ICF specifically, we don't have any dye for ICF. There's no dye which goes only into the ICF. But if you know total body water and ECF, you can subtract both and then you can calculate ICF. Interstitium and plasma. Plasma. To measure plasma, can I use something which will occupy plasma? Yes, I can use plasma proteins. Albumin, radioactive iodine, iodine 125, dye called Evans blue. All this can be used to measure the plasma compartment. For interstitium, there is no specific agent. If I know ECF and plasma, I can subtract and I can calculate interstitium. This technique is called indicator dye dilution technique, and that is how we are able to measure all the different body fluid compartments. Yes, excellent, Dr. Rahul. Can you answer this question? Which is that method used to measure volume of distribution of body fluid compartments? What is the answer? Just now we saw. Yes, indicator dye dilution method. You are right. Indicator dye dilution method. Answer C. What about the other options? What is Ketty method? This is used to calculate cerebral blood flow. What is Fick's principle? For measurement of cardiac output. Answer for this question is indicator dye dilution technique. What is the answer for this question? Yes, excellent, all of you. This one, can you try? Which is the major cation in ICF? Excellent, Dr. Rids, Rashi, Shristi, Aparajita, Afreen, Gina, Monica, Anvesha. Correct answers. This one, can you try? Yes, most of you have a tendency to go with sodium without reading the question properly. Major cation in ICF. The question is major cation in ICF. It is not ECF. If the question was major cation in ECF, then it would be sodium. Which is the major cation in ICF? It is potassium followed by magnesium. So answer for this question is potassium. Since potassium is not there in the option, we will go with the second best answer, magnesium. Look into the question properly. Major cation in ICF, inside the cells, it is potassium. Since potassium is not there, yes, see, many of you have given the answer B. Those of you who said B, be very careful. You are the people who will make silly mistake. Read the question properly. You know the answer, but you're doing a silly mistake. Major cation in ICF, it is not ECF. If it is ECF, then it would be sodium. Just for a completion, what would be the major anion in ECF? Chloride. Major anion in ICF? Proteins and phosphate. What would be the pH in ECF? 7.4. What would be the pH in ICF? 7. So these are the major differences between ICF and the ECF composition. A word about daily water intake and daily water output. How do you take in water? We drink water. Approximately we drink 2100 ml of water. As a result of metabolism, our body itself releases another 300 ml. So 2100 plus 300 is 2400 ml. This is the total daily water intake. What is the daily water output? In sweat, we lose 100 ml. In feces, we lose another 100 ml. In urine, we use 1400 ml or 1.4 liters. There is an insensitive water loss. What is insensitive or insensible water loss? Which you can't sense, you are not aware of. The water loss that happens through the lung. Water vapor, you can't see that. 350 ml. Water loss through the skin by the process of convection, radiation and all. Through the skin, there is another 350 ml of water loss. That also you can't sense. So, insensitive water loss or insensible water loss comes up to 350 plus 350. You add all this, you will get total water output of 2400 ml. The question is, an intern wants to measure the IV fluid that has to be infused to a patient. So he is taking a history of urine output and how much was the sweating for the patient, did the patient pass stools, all this he is asking. How much would he contribute to or how much would he add in his calculations as insensible water loss? How much is the insensible water loss? Yes, 350 plus 350 is 700 ml is the insensible water loss. So whenever we calculate IV fluid for patients admitted in the wards, you have to ask the patient for sweating, passage of stools, passage of urine, all this you can measure, and or as a routine you have to add another 750 ml because that 750 ml is the insensible water loss by the patient. That also you have to replace. That is the purpose of learning about insensible water loss. Yes, Dr. Muskan Agarwal, major anion in ECF. Major anion in ECF is chloride, doctor. Chloride. The concept of osmolarity and osmolality. Highly confusing topic. Most of the time we skip this topic thinking that it is very difficult. But from exam point of view, whatever is going to be asked in your exam, let me convey it to you in the easiest possible way. First, let's learn about few terms here. What is 1 mole? 1 mole means molecular weight in grams. For example, let's say this is 1 mole of any substance. I am going to take 1 mole of any substance. This is a weighing machine. I will place this on the weighing machine. Now this weighing machine will give me a value, a weight in grams. Now this weight is called as molecular weight. So what is molecular weight? The weight of 1 mole of any substance is called molecular weight. You take 1 mole of sodium chloride, you place it on the weighing machine, it will read 58.5 grams. So 58.5 grams is the molecular weight of sodium chloride. Likewise, you take any mole of any other substance, you place it on the weighing machine, the weight is called molecular weight. So that was about the definition of molecular weight, which is nothing but the weight of 1 mole of any substance. What is osmole? Now listen. Osmole means an osmotically active particle. Now for example, let me take 1 mole of sodium chloride. This is 1 mole of sodium chloride. This is 1 liter of water. Now I am going to dissolve 1 mole of sodium chloride in 1 liter of water. In water, this 1 mole of sodium chloride splits into 2 osmotically active particles. What are these particles? Sodium and chloride. So whenever I take 1 mole of sodium chloride and dissolve it in 1 liter of water, it will split into its osmotically active particles. So there are 2 osmotically active particles here. So how many osmoles are there? Sodium chloride, 2 osmoles. So 1 mole of sodium chloride, when you dissolve in 1 liter of water, it gives you 2 osmoles. Now you understand the difference between mole and an osmole. When it is outside, you can call it as a mole. But once you dissolve 1 mole of any substance in 1 liter of water, it will split into its respective osmoles. So there are 2 osmoles here. Likewise, 1 mole of calcium chloride. What is calcium chloride? CaCl2 is calcium chloride. This is 1 mole of calcium chloride. I'll dissolve this in 1 liter of water. Now see, it has split into CaCl2, 1 calcium and 2 chloride. How many osmoles are there? 1, 2, 3 osmoles are there. So when you dissolve 1 mole of calcium chloride in 1 liter of water, it will split into 3 osmoles. This will apply for salts but not for sugars. For example, you take 1 mole of any sugar, dextrose is a sugar, and you dissolve in 1 liter of water. It will stay as 1 osmol only. I'll show you. If this is 1 mole of dextrose, dextrose is a sugar, and you dissolve this in 1 liter of water, this will stay as 1 osmol only. Salts, they have ions to split. Sodium, chloride, calcium, chloride, all that. But sugars, they don't have ions to split. So they stay as 1 osmol itself. Salts will split, but sugars, they stay as such. This rule is not only for dextrose. For any other sugar like glucose or sucrose, any other sugar, 1 mole of any sugar, when you dissolve in 1 liter of water, it will stay as 1 osmol only. So that was a quick learning of the differences between mole and osmol here. What is the formula to calculate normal plasma osmolarity? Please take down this formula. To calculate normal plasma osmolarity, 2 times sodium plus 0.055 times glucose plus 0.36 times blood urea nitrogen. This is the formula to calculate normal plasma osmolarity. Can you think of this? Blood and or plasma has so many substances present in it. But why are we using only sodium, glucose and urea to calculate osmolarity? Actually, how should you calculate osmolarity? If suppose this is blood and you want to calculate osmolarity. What is the meaning of osmolarity? Concentration. Osmolarity means concentration. You want to calculate the concentration of blood. Blood has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc., etc., etc. Like this, 100 substances are present in the plasma. If you want to calculate the blood osmolarity or plasma osmolarity, you should calculate the concentration of all the 100 substances present in your blood. But we are not doing this. We are only using sodium, glucose and urea. Why is it? This is because of all the substances, only sodium, glucose and urea contribute maximum to plasma osmolarity. If plasma osmolarity is 300 milliosmoles per liter, out of this almost 297 milliosmoles per liter will be contributed by sodium, glucose and urea. So that is why we use only these substances. We choose these substances as the major substances. If you want, you can sit and calculate all the 100 substances, but it is a very difficult process. Practically not possible. So you create a formula, it should be easy. So that is why we use only sodium, glucose and blood urea nitrogen. What is the difference between these two terms? Osmolarity, R-I-T-Y, and osmolality, L-I-T-Y. Rity and lity. Is it same? No, it is not the same. Osmolarity is number of osmoles present in 1 liter of the solution. And osmolality is number of osmoles present in 1 kilogram of the solvent. Lity and rity are not the same because liter and kilogram is not the same. Liter is different, kilogram is different. For example, I take 1 liter of water and let me dissolve 1 mole of sodium chloride into 1 liter of water. What is the osmolarity of the solution? Look at this. I take 1 mole of sodium chloride and dissolve this in 1 liter of water. How many osmoles are there? 2 osmoles are there. So what is the osmolarity? Number of osmoles present in 1 liter. How many osmoles are there? 2 osmoles are present in this 1 liter. So the osmolarity is 2 osmoles per liter. So that is how the osmolarity becomes 2 osmoles per liter. Now let's calculate the osmolality for the same. I dissolve the same 1 mole of sodium chloride into 1 liter of water. What is osmolality? Now also it is 2 only. 2 osmoles per kilogram. So osmolarity and osmolality are same. Then why am I saying that it is not the same? I'll tell you one thing. Have you noticed that in your physiology textbooks or physiology exams, in one question you will see what is the normal plasma osmolarity. In another question, you will see what is a normal plasma osmolality. Even for the value, even in Genong, if you see, in one page they would have written like this. The normal plasma osmolarity is 300. In the second page, you will see normal plasma osmolality is 300. If osmolarity and osmolality is not the same, then why are we interchanging these terms? We can interchange these terms if the underlying solvent is water. If you dissolve 1 mole of sodium chloride in 1 liter of water, here also it is 1 liter of water. If the underlying solvent is water, for water, 1 liter is equal to 1 kilogram. If it is water, liter and kilogram both are same. And remember, only for water it is the same. For any other solvent in the universe, 1 liter and 1 kilogram is not the same. Only for water, 1 liter and 1 kilogram are same. For example, this is 1 liter of water. I'll put it on the weighing machine. This will exactly give me a weight of 1 kilogram. You take 1 liter of ethanol, which is a hand sanitizer. You take hand sanitizer, 1 liter, and you weigh it. It will not give you 1 kg. It will give you only 0.7 kg. For any other solvent other than water, 1 kg is not equal to 1 liter. So if I am talking about water, I can very well say osmolarity, osmolality both are same. If I dissolve 1 mole of sodium chloride in 1 liter of water, osmolarity is also 2, osmolality is also 2, because 1 liter of water is equal to 1 kilogram. That is why in physiology, biochemistry, or even in medicine, whenever you talk about your underlying body fluid, in medicine we always talk about our body. Our body is made up of water. So that is why in physiology, biochemistry and medicine, in the entire MBBS curriculum itself, you can very well interchange osmolarity and osmolality. Even when you have learned your BDS, dental, or when you are preparing for your MDS, in our medicine itself, we can interchange osmolarity and osmolality, because underlying solvent is water. We are talking about the human body. So that is how osmolarity and osmolality became the same. What is osmolar gap? Osmolar gap is measured osmolality minus calculated osmolality. This is called osmolar gap. What does this indicate? How do you measure osmolality? You take blood and you put it into the machine, osmometer, and this will calculate all the substances present in your blood, and this will give me 300 milliosmoles per liter. This is the measured osmolality. What is the calculated osmolality? Sometimes we have to sit and calculate using the formula. When you calculate using the formula, what is the formula? Sodium plus glucose plus blood urea nitrogen. This is the formula. We already saw the formula. When you use only sodium, glucose and urea, we get a value of 297 milliosmoles per liter. If the machine is doing it, you get 300. But if I sit and calculate, I am getting only 297. What is the difference due to? The machine is taking whole blood. It is calculating all the 100 substances in the blood. It is giving me 300. But if I sit and calculate in the formula that I use, only sodium, glucose and urea. Only 3 substances I am calculating. That's why I am getting 297. If you also sit and calculate all the 100 substances, you will also get 300. So that is the difference between measured osmolality and calculated osmolarity. So 300 minus 297 will give me a 3 milliosmole per liter. What does this 3 milliosmole per liter indicate? Yes, the substances other than sodium, glucose and urea. So this indicates the presence of osmotically active particles other than sodium, glucose and urea. 297 contributed by sodium, glucose, urea. So the remaining 3 indicates the presence of all other substances other than these 3. So that is about osmolar gap. Over to osmotic pressure. What is osmotic pressure? When there are 2 compartments separated by a semi-permeable membrane. So these are 2 compartments separated by this semi-permeable membrane. In one compartment, you have plenty of water, and in the other compartment, you have plenty of salts. How will this water move now? This is compartment 1 and compartment 2. Water is going to move from 1 into 2. What is this process called as? Yes, osmosis. So what is osmosis? Movement of water from its higher concentration to its lower concentration is called as osmosis. Movement of water from its higher concentration to its lower concentration is osmosis. Now just imagine I am going to take a big weight and I am going to press on this salt side. What will happen if I press on the salt side? Movement of this water will stop. Osmosis will stop. So that pressure which is required to stop osmosis is called osmotic pressure. That pressure which is required to stop osmosis. If I apply a pressure, this movement will stop. So that pressure which stops osmosis is called as osmotic pressure. Yes, can you do this calculation? All of you, can you just try? What is the answer for this? How do you solve this? What is the calculated osmolarity of a 1 liter solution containing 1 millimole sodium chloride, 4 millimole of potassium chloride and 2 millimole of calcium chloride? Excellent, Dr. Rahul Chandrasekhar. Very good. This was exactly a repeat question also. How do you do this? If you dissolve 1 millimole of sodium chloride, this is 1 millimole of sodium chloride. If I dissolve in water, it will split into 2 osmoles. So this splits into 2. This will give you 1 into 2 is 2 milliosmole. Let's take 4 millimoles of potassium chloride, KCl. This will also split into 2. 4 into 2 is 8 milliosmole. 2 millimole of calcium chloride, CaCl2. So if I take calcium chloride, CaCl2 and dissolve in water, this will give me calcium and 2 chloride. 3 osmotically active particles into 3. 2 into 3 is 6 milliosmoles. So totally this is 16 milliosmoles. Excellent. Dr. Anand Sinha, right. Both of you are right. This is how we solve problems based on osmolarity and osmolality. Over to types of fluids. What are the different types of fluids? The purpose of learning this different type of fluid is usually in casualty or emergency. When you are posted as an intern there, you receive a patient on an emergency situation. You won't know which fluid to be infused for which type of patients. First of all, you should know what are the fluids. Then only you will know the indication for these fluids. What is an isotonic fluid? All fluids which have same concentration as your plasma, they are called isotonic. If plasma concentration is 300, any fluid which has 300 is called isotonic. Hypotonic means all fluids which have less concentration than your plasma, they are hypotonic. All fluids which have more concentration than your plasma, hypertonic. What are the examples? Examples for isotonic fluids: 0.9% saline, ringer lactate, RL, 5% dextrose in water. What is the indication for these solutions? To whom will you give this? If a patient is losing his plasma or blood or body fluid and you just want to replace the body fluid, you can give him a fluid which has same concentration as your body. When will you lose your body fluids? When you are bleeding, hemorrhage or vomiting or diarrhea, burns, trauma. In all these conditions, you just want to replace the body fluid, so you give them a fluid which is same as your body fluid. Hypotonic fluids. Example, 0.45% saline. 0.9 saline is isotonic, so 0.45, half of that would be hypotonic. We use them in hypernatremia patients. If a patient is already having more salts and there is an indication for fluid transfusion also, now you can give him a fluid which has less salts. So in hypernatremia patients, hypotonic fluid. Hypertonic fluid, example 3% saline and 10% dextrose. When will you use this? In hyponatremia patients. Already the patient is having less salt. There is an indication for fluid transfusion also. Now safely you can give him a fluid which has more salts. So that is about the tonicity of these fluids. Isotonic, hypotonic and hypertonic fluids. Next, let's see the change in the Darrow-Yannet diagram on transfusion of these fluids or in any other clinical condition. What is the change in the Darrow-Yannet diagram? Let's do that. First of all, let's read this diagram. This diagram is called the Darrow-Yannet diagram, which has x-axis and y-axis. On x-axis you can see the volume of ECF and ICF. On y-axis the osmolality, concentration of ECF and ICF. The solid colors that you see are actual ICF and ECF. How much is the ICF? 2/3, the pink one. How much is the normal ECF? 1/3, the blue one. This is the normal ICF and ECF. First, let's do few basic points in Darrow-Yannet diagram. There are 6 conditions that we are going to learn. 3 gain of fluid conditions and the other 3 loss of fluid conditions. First, I'll tell you few rules to read this diagram. The first rule is that whenever you read this diagram, you look only into the changes in ECF. No need to look at the ICF at all, because whenever there is any loss of fluid or gain of fluid, it is always from the ECF. For example, if I infuse 1 liter of saline into the patient, how will you infuse fluid? You take an IV line and you inject the fluid. When you inject an IV line fluid, this fluid will go into the blood. Blood is ECF. Or if I am vomiting, I am losing my body fluid. That is also from ECF. So any gain of or loss of fluid will always be first from the ECF only. So look only into the ECF. And ICF will make changes according to the ECF. Anyways, you see, ICF will change according to the ECF. Finally, after equilibrium, ICF and ECF both will reach the same concentration. So don't worry about ICF. Anyways, in all diagrams, somehow it will manage and reach the equilibrium. Don't get confused by looking at the ICF. The second rule is that whenever there is any gain of fluid condition, on x-axis, volume of ECF increases. Yes, if the body is gaining a fluid, volume of ECF will increase. And vice versa also is true. If there is any loss of condition, on x-axis, volume of ECF will decrease. Yes, if you lose a fluid, your ECF volume will decrease. So that is one hint to identify the diagram. Any gain of conditions, ECF volume increases, and any loss of conditions, ECF volume will decrease. So that is about gain of and loss of conditions. And number 4, if it is an isotonic fluid, in the case of isotonic fluids, any isotonic fluids, whether you gain an isotonic fluid or you lose an isotonic fluid, there is not going to be any change on the y-axis. No change on y-axis. Why is that? Because isotonic fluids, you gain an isotonic fluid or you lose an isotonic fluid, it is a fluid which has same concentration as your body. So it will not change your body concentration. That is why in the case of isotonic fluids, no change on y-axis. So these are all few basic rules before you read this diagram. Now let's move on to the actual diagram. The Darrow-Yannet diagrams. I'll show you 6 diagrams. With these rules, let's do that. The first diagram, gain of isotonic fluid. Example, infusion of isotonic saline. Anyway, it is a gain of fluid. Whenever you gain a fluid, ECF volume on x-axis increases. That is why the dotted line has come out. Solid color is the normal ECF ICF. Dotted line is the change. Dotted line has moved out. So whenever there is a gain of isotonic fluid, ECF volume increases. Isotonic fluid. It's an isotonic fluid. If you gain a fluid which has same concentration as your body, there is not going to be any change on the y-axis. So y-axis no change. The solid line and the dotted line are all same. It's a gain of isotonic fluid. Number 2. Gain of hypotonic fluids. Example, infusion of 0.45 saline, water intoxication, SiADH. Anyway, it's a gain of fluid. If you are gaining a fluid, ECF volume should increase. It's a gain of hypotonic fluid. Hypotonic fluid means fluid which has less concentration than your body. What happens in SiADH? What is actually SiADH? Syndrome of inappropriate ADH secretion. Inappropriately, so much of ADH is secreted. What is the role of ADH? Antidiuretic hormone. It will cause water reabsorption into the blood. The more you reabsorb water into the blood, blood becomes dilute. Blood becomes dilute. Water has gone into the blood. So what will happen to the water that is in urine? There is no water going out in urine. All water has gone into the blood. So the blood becomes dilute. So what will happen to the ECF concentration or blood concentration? Blood concentration decreases. ECF concentration decreases because it is getting diluted. So x-axis volume is increasing. On y-axis ECF concentration decreases. This is gain of hypotonic fluids. Let's go on to the third condition. Gain of hypertonic fluids. Infusion of 3% saline, 5% saline, 10% dextrose, excessive salt and water intake. You take more of salt water. Anyway, it is a gain of fluid. Whenever you gain a fluid, ECF volume increases on x-axis. You are gaining a hypertonic fluid. You are adding more salts into your body. So what will happen to your ECF concentration? Definitely it will increase. That is why volume has increased on x-axis. Concentration also has increased on y-axis. Dotted line has gone up. This is the change in Darrow-Yannet diagram in gain of hypertonic fluids. Next, let's move on to the laws of conditions. Laws of isotonic fluids. When will you lose an isotonic fluid? You are losing a fluid which has same concentration as your body. Hemorrhage, vomiting, diarrhea. Anyways, it's a loss of fluid. So ECF volume decreases. It's an isotonic fluid. If you lose an isotonic fluid, there is not much change in the concentration. So the dotted line and the solid line on y-axis remains same. Let's move on to the fifth diagram. Laws of hypotonic fluid. Anyways, it's a loss of fluid. So x-axis volume will decrease. You are losing a hypotonic fluid. What is a hypotonic fluid? A fluid which has less concentration than your body. Sweat. Sweat is a hypotonic fluid. See, generally we think that you taste sweat, it is salty. But it doesn't mean that sweat has more salts than your blood. It is salty only, but it has less salts than your blood. So sweating is a hypotonic fluid. Diabetes insipidus. What is diabetes insipidus? This is a disease where you have deficiency of ADH. What is the role of ADH? Water reabsorption into the blood. Water is reabsorbed into the blood. This is the role of ADH. But if there is deficiency of ADH, water is not reabsorbed into the blood. If water is not reabsorbed into the blood, water goes in urine. That is why diabetes insipidus patients have so much of water in urine. If you lose so much of water in urine, water goes out in urine, ECF concentration increases. So that is why y-axis, the dotted line has gone out. Number 6. Loss of hypertonic fluids. Anyways, it's a loss of fluid. So definitely x-axis ECF volume will decrease. It's a loss of hypertonic fluid. You are losing a more concentrated fluid from the body. What is Addison's disease? Addison's disease is a condition where adrenal deficiency. See, Addison's means ADD, adrenal deficiency. What will happen in adrenal deficiency? The adrenal hormone aldosterone is less. What is the role of aldosterone? Salt reabsorption into the blood. This is the role of aldosterone. If aldosterone is less salt is reabsorbed into the blood. So where does all this salt go? It goes out in urine. If you lose so much of salt in urine, your body concentration decreases. That is why ECM concentration has decreased. So these are 6 different conditions in Darrow-Yannet diagram. 3 gain of conditions and 3 loss of conditions. Yes, can you solve this question? One of the repeat questions. Can you give me the right answer for this? Shrishti, you want a break? Sure, doctor. We have few more topics to complete. Okay, so we'll have a 5 minutes break and we'll move to cell physiology. Since Dr. Shrishti is asking us for a break, yes, we'll have a 5 to 10 minutes break. Meanwhile, you find out the answer for this question and we will join after a short break for cell physiology. We are almost done with general physiology. Over to cell physiology after a break. Done?